Welcome back to this next video in which we are discussing the human papillomavirus entry mechanism. Okay, so we're currently discussing the endocytosis of the human papillomavirus bound to the secondary receptor. And this is quite a controversial topic. Different papers describe it using different endocytosis mechanisms, and these are the 3 endocytosis mechanisms that we're going to go over. Okay, so remember I said that there were different possible explanations for this. Potentially different types of human papillomavirus use different secondary receptors and then different endocytosis mechanisms, or maybe they all use the same secondary receptor, it's just that this secondary receptor can be endocytosed in multiple different ways. So we're going through all these different possible endocytosis pathways, and we've begun with the major, best understood one, which is clathrin-mediated endocytosis. So to describe what we've seen so far, remember this process does not go to the cargo. It occurs at portions of the membrane where there is a lot of the special phospholipid PIP2. And the reason it occurs at these portions of membrane, and it has to be in the inner leaf that this PIP2, is because the adaptor proteins binds to PIP2. So where there are lots of PIP2 molecules in the inner leaf that are the phospholipid bilayer, you get adaptor proteins binding there. This causes the membrane to curve inwards to form a little pit here, and these adaptor proteins also bind to the clathrin trischelia, so they recruit clathrin trischelia here, and the clathrin trischelia begin to polymerize into their spherical structure, and this is then known as a clathrin-coated pit. This progresses and you get a deeper pit forming here, like so. Meanwhile, you're also getting cargo recruited into this area which is being endocytosed because receptors that are going to be internalized have internalization motifs on their cytoplasmic aspect, and as they come into this portion of the membrane, just because they're diffusing about, they will get stuck here by binding to the adaptor proteins, which have binding sites for these different internalization motifs. And as I said, there are lots of different internalization motifs, and the different adaptor proteins will have different internalization motifs that they can bind to. So when we have our secondary receptor with the virus bound to it, there are 2 possibilities. Either the secondary receptor has a permanently exposed internalization motif and would have been endocytosed whether the virus was bound to it or not, and the virus is just hitchhiking a lift, or when the virus binds, it causes a change in the conformation of the secondary receptor, exposing the internalization motif, and then the whole complex is internalized or targeted into these clathrin-coated pits. So that's the point that we've got to so far. Now, you might think that, how is this membrane portion going to bud off? Well, maybe it just occurs because the clathrin polymerization just continues on, completes up the sphere and cuts off this portion of membrane from the plasma membrane. Maybe that's the way that this pit is going to bud off to become an endocytic vesicle. Well, that's not the answer, I'm afraid. The clathrin polymerization doesn't have the power, it doesn't have the energy to break the membrane, it doesn't have the energy to cut the final bit of membrane. It needs more help. It's certainly going to help in this cutting of the membrane, but there's a more powerful process that's going to cut off this pit of membrane that we've got here, this invagination of membrane that we've got here from the plasma membrane. And this involves a protein called dynamin. So let's now talk about dynamin, and I think I'll go for orange when we're discussing dynamin. So we'll do dynamin down here. So the next, whoops, where's my pen? The next process we're going to discuss, and why have I got such a thick pen, is dynamin. Okay, so dynamin is a protein that is capable of binding to this portion of membrane here at the top of the clathrin-coated pit. So it's this bit of membrane that we need to close up. We need to close up this tube that links this cavity to the extracellular fluid. We need to bring this close together and cut it off and join this bit back to this bit. And this is what dynamin is going to do. It is going to bind together, it's going to oligomerize together and form a spiral that is going to go around this, I'm trying to figure out what you should call it, the neck of the pit, if you like, this connection between the plasma membrane and the membrane of the clathrin-coated pit. So let me draw another little picture of this here. So let's now just draw the membrane as a single line. So rather than drawing the membrane as a phospholipid bilayer, I'm going to draw the membrane as a single line, like so. And here is the neck of the clathrin-coated pit, and this is where dynamin proteins are going to oligomerize around here to form a spiral. So let me now show them forming a spiral. So I'll represent each dynamin protein with a dot, like so. So what's going to happen is dynamin proteins around the neck are going to oligomerize together, like so. So they can bind to this very curved portion of the membrane. So that's what dynamin proteins are capable of doing. They can bind to very, very curved portions of membrane. And the neck of this clathrin-coated pit that we've got here, this is very, very curved, and this is ideal for the dynamin to bind here. So it forms a spiral, and this will go round the back of the neck that we can't see, and then it will come back here. So the dynamin proteins form a spiral around the neck of the clathrin-coated pit. And just to complete up this picture, I would like to put some clathrin on here. So with this very thick purple here, this is just representing the fact that this membrane is surrounded by the adaptor proteins bound to the PIP2, and then the clathrin around that. And in fact, let's also add on the secondary receptor onto here. So here we have our secondary receptor bound to the virus particle. So this is where we are at the moment. So we've got our clathrin-coated pit here with the secondary receptor and the virus particle bound in it. And then the way that this is going to be clipped off from the plasma membrane is that dynamin proteins are going to oligomerize into a spiral around the neck of the clathrin-coated pit, like so. And then all of the dynamin proteins, they are capable of harvesting energy. What this spiral is going to suddenly do is it's going to constrict. It's going to suddenly change conformation and become much narrower. It's going to constrict so that the diameter of the spiral is hugely decreased. And of course that will constrict the neck so that it's going to be clipped off, so that the clathrin-coated pit here will be clipped off from the plasma membrane. Now, where do the dynamin proteins then get the energy for doing this from? They get it from the hydrolysis of Gtp. So let's talk about this now. So dynamin proteins, they are all actually little enzymes that are capable of hydrolyzing Gtp. So they're all GTPases. Now, all of these are going to bind a GTP molecule, and then synchronously, once the spiral has completely assembled, as much as it's going to assemble, they all start hydrolyzing GTP. So GTP is very similar to ATP, except that the organic base, instead of being adenine, is going to be guanine, so guanosine triphosphate. So you can cut off the third phosphate group and hydrolyze this to guanosine diphosphate, GDP, and a phosphate group. And when you do that, it releases energy. So all of these dynamin proteins, synchronously, they hydrolyze the GTP to GDP and inorganic phosphate. This gives them the energy to then change conformation. The whole spiral changes conformation and it constricts. So it will constrict this neck down, and it will also elongate the neck. So if you imagine what is going to happen to this spiral, if it's going to reduce its diameter, then its length has to go somewhere, so it's going to become longer upwards. So let me just draw out the change that is going to occur to the neck of this clathrin-coated pit. So it's going to go from looking like that to maybe looking like this. So the neck has become much thinner and much longer, and that's because the dynamin spiral has constricted. So now let me draw on the dynamin spiral again onto this picture. So here they are. So now the spiral has a much smaller diameter, and because it's still got the same number of dynamin proteins in it, that means that it has to be much longer. If its diameter's decreased, then where have all the dynamin proteins gone? They've gone into lengthwise length. So the spiral is much longer but much thinner, so it's constricted down the neck, and this is what eventually clips this off. It clips this clathrin-coated pit here off from the plasma membrane, and then what you will have is the clathrin will complete its polymerization so that you have the complete clathrin shell around this membrane. So what you end up with at the end of all of this, so this goes to this, and then after that what you end up with is, here is the plasma membrane, and then you've got a separate vesicle inside the cytoplasm of the cell that will have clathrin surrounding it. So let me put the clathrin in here in purple, and of course I'll actually draw this a little bit better. So we'll have I'll draw the adaptor proteins on as well as the clathrin, just because they're going to be important in the next bit of the story. So here are the adaptor proteins, which will all be bound to PI-P2 molecules in the membrane of this vesicle that we've now budded off here, and then we'll have the clathrin proteins surrounding those, bound to those. So here is the clathrin protein, and all the way around you've got the clathrin sphere that's assembled here. Okay, so that is what we overall end up with in the process of clathrin-mediated endocytosis. And of course, in the membrane of this vesicle that we've got here now, we will have the secondary receptor with the virus particle bound to it. Okay, so what have we now ended up with? Let me go back to the thin pen and let's write some stuff down. So this structure that we've got at the moment is now called a clathrin-coated vesicle, CCV for short. So this is a clathrin-coated vesicle. So the clathrin-coated pit has now been pinched off to form a clathrin-coated vesicle, which is in the cytoplasm of the cell. And that makes complete sense that it's called a clathrin-coated vesicle, because it's a vesicle that is covered in clathrin. Now, this clathrin-coated vesicle cannot yet go into the endocytic pathway. It can't go into the endocytic pathway until the clathrin coat has been removed. So the next step that has to happen is uncoating. So next step along is the uncoating process. So the clathrin buckyball will have to disassemble now, and also the adaptor proteins are going to come off from the membrane of the clathrin-coated vesicle. And this process isn't completely understood, but we think what actually must happen is that the PI-P2 molecules that were in the inner membrane, the inner leaflet of the membrane that we pinched off here, we think that those are destroyed. They're turned into a different type of phosphatidylinositol molecule, and that's the way that the adaptor proteins come off the membrane, and then the clathrin buckyball has to come off as well. So uncoating occurs, and then of course what you end up with is the vesicle here that is now ready to go into the endocytic pathway. So this is what we would now call an endocytic vesicle, and this will now contain all of the receptors that were recruited into this region that was having the clathrin-mediated endocytosis occur there. So this is an endocytic vesicle, and of course the one that we're very interested in is that secondary receptor bound to the virus particle, which I'll just draw in here. So here is our secondary receptor, and then the virus particle in evil red is here. Okay, so the endocytic vesicle with its cargo present inside it is now going to go into the endocytic pathway. So I'm going to discuss the endocytic pathway now, following my discussion of clathrin-mediated endocytosis, just to complete up the story. We'll then go back and discuss caveolin-mediated endocytosis, which is also going to lead into the endocytic pathway. Indeed, all of the different endocytosis pathways, they're overall going to give internal membrane-bound vesicles, like so, which can then enter the endocytic pathway. So, let's have our yellow colour. So I'll just put a title. We're now going to discuss what is called the endocytic pathway, which is what happens to endocytic vesicles. And it's worth remembering that this is physiology. This isn't pathology. We're going to discuss it so that we can discuss the pathology, but this is what would happen to an endocytic vesicle normally when clathrin-mediated endocytosis has created it. What happens next? Where does all the cargo that's potentially inside of this endocytic vesicle go? It goes into this pathway. It's just that the virus bound to this secondary receptor is going to hijack this system and therefore is going to follow this pathway. So the endocytic pathway. So I'm just saying that so that you know that this isn't just pathology, this is physiology that's being hijacked by this virus. Okay, so we've now got our endocytic vesicle from, in this case, the process of clathrin-mediated endocytosis. Where does it go next? So lots of endocytic vesicles all join together to form something called an early endosome. So drawing a little picture here, let's say this is the cell membrane of our cell. Let's say, I'll do in blue, that this is an endocytic vesicle. This may be another endocytic vesicle, so we've got lots of little endocytic vesicles here. What's going to happen is lots of endocytic vesicles join together to form something called an early endosome, which I'll do here in green. So this structure is going to represent an early endosome, and it has these little tubules coming off like so, which I'm drawing here. Okay, so this structure is an early endosome, which for short can be abbreviated down to EE, like so. So this is an early endosome. Now, early endosomes, although they're formed from the endocytic vesicle, they're going to have very different properties to endocytic vesicles. In particular, once endocytic vesicles start fusing together to form an early endosome, the pH inside the early endosome starts going down, and this is because there are all sorts of things going to be implanted into the membrane once these things start fusing together to form an early endosome. So lots of endocytic vesicles join together to form an early endosome, and then as you continue to produce more endocytic vesicles, they will join this thing to make it bigger and bigger, and this thing has very different properties to the endocytic vesicle because lots of proteins have been added into the membrane here. In particular, proteins that are going to sequester protons from the cytoplasm into the lumen of the early endosome have been added into the membrane, and that means that the pH inside this thing is quite low. And that's going to be important later on when we want to discuss the virus going through this pathway. So the early endosome has a low pH. Now, before I tell you what those tubular structures coming off the side of the early endosome are for, let me tell you what eventually happens to early endosomes. So they gradually change more and more. It's a gradual process. Endocytic vesicles fuse together to make this thing. More and more endocytic vesicles come to make this thing bigger and bigger, and gradually it's changing in properties. Its pH is getting lower and lower is one of the key things that is changing, and gradually initially it's going to have these tubular structures coming off the side of it, and we'll see what their function is in a moment, but gradually it loses those, it gets fewer and fewer of those, and the ones that it still does have are smaller, like so, and it becomes what we would call a late endosome. So maybe it just has one little tubule maybe left there, and gradually even those ones are going to disappear as it becomes a later and later endosome. So it gradually matures this endosome into what we would call then a late endosome, which doesn't have these tubular structures coming off anymore. And then what finally happens is the late endosome fuses with cytoplasm structures called lysosomes. So I'll put these in here. So this can represent a lysosome. And lysosomes are full of loads of very, very powerful and very dangerous enzymes, which overall are called lysozymes. So lysozyme is just a name for an enzyme inside a lysosome. There are loads of different lysozymes. They're all extremely powerful and extremely dangerous, and they're very, very good at breaking down biological molecules. So what's going to happen is these are going to fuse with the late endosome eventually and put all their incredibly dangerous contents into the lumen of the late endosome, and this is so that you can break down all of the stuff that's in the late endosome. So remember, overall, the motivation for what's happening here, we are bringing in stuff from the outside of the cell membrane, stuff that the cell wants to use. It might contain deliveries for the cell. So they're brought into these early endosomes, the early endosome goes into a late endosome, and then eventually the lysosomes fuse with the late endosome so that we can break down all of the stuff that we're bringing in into smaller molecules that the cell can then use. That's the motivation for what is happening here. The cell is getting deliveries, and we are then processing the deliveries by using all of these incredibly dangerous enzymes in the lysosomes. Okay, now the endocytic pathway, this is the major arm of the endocytic pathway, what we've just done. This is the major way that you get your delivery and then you process it. However, these tubules are important as well, so deliveries that you are bringing in, you don't necessarily want to be broken down by the lysosomes. In particular, often, let's say you brought in some cargo that's bound to a receptor. Now, I'll use this picture as an example. I know this picture is representing the secondary receptor with the virus bound to it, but let's pretend that this is a physiological example rather than a pathological example. Let's say this is some receptor with some cargo that we want to process here that's bound to it. We've brought this into the early endosome. If we let this entire thing go through to the late endosome and then be exposed to the lysosomes containing their lysozymes, the whole thing will be broken down. The cargo and the receptor will be broken down. That's not completely efficient, because actually we only want to break down the cargo in some cases. We'd like to keep the receptor and recycle it and use it again. So ideally, what would happen is we'd break the cargo away from the receptor, we'd keep the cargo in the endosome, we'd let it be broken down by the lysosomes, but we'd traffic the receptor back to the cell membrane, and that is indeed what happens. And this is the whole point of the pH getting lower and lower in endosomes. And by the way, the pH is even lower in the late endosomes compared to the early endosomes. So as an endosome matures, so that's what this whole process is called here. This is endosomal maturation. So this is maturation here. As the endosome matures, the pH gets lower and lower, more and more acidic. And the whole purpose of that is that when you have a change in the pH, it often changes the conformation of proteins and can cause molecules that might have once bound to each other to cleave apart. So that's the idea, that you bring in the receptor with cargo in, the pH gets gradually lower and lower. Eventually, if the pH changes enough, it will cause the cargo and the receptor to break apart. The cargo will then be in the lumen of the endosome. You can leave it in there, awaiting destruction by the lysozymes or processing by the lysozymes. Meanwhile, let's take this receptor that's in the membrane of the endosome and traffic it back to the cell membrane, which is what these tubules are for. These are for budding off more vesicles. So off these tubules, little vesicles can bud off, little bits of membrane can bud off the endosomes, and these are going to deliver receptors back to the cell membrane. So they can either go directly back to the cell membrane in a very fast process, or they can go via another membrane-bound structure called a recycling endosome, which I'll put here. So this can represent a recycling endosome, again another membrane-bound organelle. So this little vesicle that's come off our endosome, it can either go directly back to the cell membrane, or it can firstly go to this structure called a recycling endosome, and then it can bud off the recycling endosome and go back to the cell membrane. Now, what's the purpose of a recycling endosome? Well, sometimes you don't necessarily want to put the receptor back in the membrane straight away. Maybe you only want some of this cargo. Imagine that you only want a little bit of this cargo that this receptor's brought in. You don't want to destroy the receptor, but you don't want to put it straight back in the cell membrane, otherwise you'll get too much cargo. So you can then store it in the recycling endosome for a while until you're then ready for more of this cargo, and then you can put it in the cell membrane. So the recycling endosome is effectively a store for receptors that might bringing cargo that you don't actually need at the moment, but you want to keep the receptor for when you do need some cargo. So, from the endosomes then, bud off vesicles that can go either straight back to the plasma membrane directly, or they can go to the recycling endosome. Another place that they can go is to the Golgi apparatus, and then the Golgi apparatus might be able to make a few modifications to the receptor and then send it back to the cell surface. So I'm wondering where I can now put this on my picture. So I'm just going to extend my picture a little bit. So here is some more plasma membrane. So the cell's been extended somewhat now. Ignore this bit. Imagine that's not there and the cell just continues on here. So drawing in another organelle here, and we'll do it in, what interesting colour can I come up with? We'll do it in this strange yellowy orange colour. So another really important organelle, of course, is the Golgi apparatus. And remember, the Golgi apparatus consists of lots of stacked membrane-bound compartments known as cisternae, like so. So this can represent the Golgi apparatus. Usually you'd have more of those membrane-bound disc compartments than I've drawn there. I've just drawn 3, but this is representing the Golgi. Now, the side of the Golgi that faces away from the centre of the cell and towards the outside of the cell, so this bit up here, this is known as the Trans-Golgi Network. And the portion that faces the centre of the cell faces where the nucleus is and where the endoplasmic reticulum around the nucleus is. That's called the cis-Golgi network. So just putting some of this on. So CGN stands for cis-Golgi network. Remember, cis means on the same side as. So this is the side of the Golgi that is on the same side as the endoplasmic reticulum. It's facing the centre of the cell where the nucleus would be with the endoplasmic reticulum around it. And I won't put the endoplasmic reticulum on at the moment. Later on in our story, it will be important to put the endoplasmic reticulum on, but we'll come to that later on. So TGN then stands for Trans-Golgi Network, and I can't really fit the Golgi in there, so I won't, and networks, I'll just put trans. So it means on the opposite side, so it's not facing the centre of the cell, it's facing the outside of the cell. Now, the Golgi network is often responsible for making proteins that are going to go to the membrane and then sending them to the membrane. Actually, I think I will put, after just saying what I've said about not wanting to put the endoplasmic reticulum on, let's now actually put the endoplasmic reticulum on. Let's do this properly. So, let's put the nucleus on, in green here. So this is the inner nuclear membrane. Remember, the endoplasmic reticulum is continuous with the intermembrane space between the two membranes of the nucleus. Remember, a cell nucleus is not just one membrane, it's a double membrane, like so. So this is the nuclear envelope, as we call it, consisting of these two membranes. So you have the inner membrane of the nuclear envelope and the outer membrane of the nuclear envelope. So this is the nucleus that we've got here. And you then have a space in between those two membranes, the intermembrane space, and this is continuous with the space of the endoplasmic reticulum. So now here comes a portion of endoplasmic reticulum, like so, and it's going to have a bit of a strange shape just because of the space that I've got. So this here is endoplasmic reticulum, and you can see how the space inside the endoplasmic reticulum, the lumen of the endoplasmic reticulum, is continuous with this intermembrane space. Indeed, the membrane of the endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope here. Okay, so here you can see that the endoplasmic reticulum is touching just about the cis-Golgi network, hence why this is called the cis-Golgi network. It's on the same side as this endoplasmic reticulum border here. So, the complete story then, the nucleus of course is where the DNA is. DNA will be transcribed to RNA, RNA will go to the endoplasmic reticulum, it will go into the cytoplasm and then often you will have the ribosomes on the surface of the endoplasmic reticulum, which will then translate the RNA into protein. And when we have proteins that we want to become receptors on the cell surface, or we have proteins that we want to secrete from the cell, those will be channeled into the endoplasmic reticulum. So there are lots of little transporters on the endoplasmic reticulum membrane which will take proteins from the cytoplasm where they've been synthesized into the endoplasmic reticulum lumen, and these are proteins that are either going to become cell surface receptors or are going to be secreted from the cell. The endoplasmic reticulum will then send little vesicles, and here is an example, to the cis-Golgi network, which will deliver these proteins that are going to become either cell surface receptors or secreted proteins to the cis-Golgi network. It'll gradually move through these membrane-bound compartments of the cis-Golgi network, these cisternae, and I do just want to write down that key word. So the compartments of the Golgi network are called cisternae. So it'll move through the cisternae of the Golgi structure here, the Golgi apparatus, gradually being processed, of course, all the way. So these proteins were modified in the endoplasmic reticulum, then they're modified in the Golgi, and then finally, once they've made it up to the Trans-Golgi network, they're then put into secretory vesicles. So here is a secretory vesicle, which will then be exocytosed. It will be fused with the plasma membrane, delivering either the protein that's in this membrane to the plasma membrane, if it's a receptor that you're going to put on the cell surface, or if it's a secreted protein, it'll be just in the lumen of this secretory vesicle and it will be released. Now, why have I just told you all of that? Well, another thing that can happen to these little vesicles that are coming off the early endosome is that they can go back to the Trans-Golgi network and be modified. They might move back through the Golgi network, potentially even go back to the endoplasmic reticulum, be modified, and then they might be put back onto the surface. So you might have modifications that you want to do. And this actually, we think, is what's going to happen in the case of the virus, but we'll come on to that later on. Okay, so it can go back and fuse with the Trans-Golgi network, and then it can make its way back through this process that I've just described here, even back into the endoplasmic reticulum here. Okay, so this is my complete picture, you'll be relieved to hear, of the endocytic pathway, and this is what usually happens to endocytic vesicles when they have some cargo within them here. So let me just summarise what I've told you. So here is the plasma membrane. We will be budding off endocytic vesicles that have receptors bound to cargo inside them, or potentially just receptors that we want to remove from the plasma membrane. Maybe we've got too many of those receptors. Maybe we want to desensitize some of our signaling pathways. They will come and fuse together to make one of these massive, great early endosomes here. Now, the early endosome changes its properties from the endocytic vesicle. The pH starts to go down, and the purpose of this is to detach cargo from receptors that we don't want to be destroyed. Those receptors can then be removed from the early endosome by membrane budding off from these tubule structures of the early endosome, and these vesicles that have come back off from the early endosome, which might just have the receptor in now, they can either go straight back to the plasma membrane, or they can go to a recycling endosome, which will store them for a while until you're ready to put them back on the plasma membrane. Alternatively, they can go back into this Trans-Golgi network over here, where they might even go back into the cis-Golgi network, or even back into the endoplasmic reticulum. So they might go back along the secretory pathway, be modified somewhat, and then they can be released again. They can make their way back along the secretory pathway in the correct direction and go back up to the plasma membrane. So that's another option for what they can do. And I only mention this because it's actually going to be important, we think, in our story. So if that doesn't happen, so if you remain in the endosome gradually matures, and then it's a gradual process. It isn't suddenly the case that it was once an early endosome and then it becomes a late endosome. It's a gradual transformation process. The pH gets lower and lower and lower and lower. The tubules gradually get smaller and smaller, and you get fewer and fewer of them. So it gradually matures into what we would then call a late endosome. So overall just refer to both of these as the endosome, and it's going through a maturation process. It's not the case that you can suddenly say, oh, it's now a late endosome. It's not a switch-like process. It's a gradual transformation, and once it's transformed enough, we would then call it a late endosome. So fewer vesicles are coming off late endosomes now, very few. And then as you get towards the end of the maturation process, none will be coming off anymore. And then what is left in this late endosome, and which has not been recycled now, that's going to then be processed by these lysozymes. So the lysosomes will fuse with the late endosome, and everything that's in there will be broken down into small biological molecules that can be used by the cell. So this is the endocytic pathway. I hope you now understand the physiology of this. We'll come on to how the virus is going to abuse this later on. To complete the clathrin-mediated endocytosis of the virus, at present we have a clathrin-coated vesicle with the secondary receptor bound to the virus in it. This is going to fuse with the early endosome, and therefore, in our pathological process, the point that we have got up to now is where here is the secondary receptor in pink there, with the virus in vivid red bound to it. I've put it on that odd angle just to avoid writing over the pH there. That's the point in the pathway that we've now got. So if I go back over here, number 7 is that the virus is now in the early endosomes, HPV in early endosome. So I have now only described how it gets there for the clathrin-mediated endocytosis process. We're going to have a break here, and in the next video we will then describe how it can get to stage 7 through the caveolin-mediated endocytosis and then the macropinocytosis process. And then we'll follow its trafficking through the endocytic pathway, how it's going to abuse all of that physiology that we've just been discussing.